Hello, everyone. Thank you for joining today's in-person seminar. Today, we are really excited to host Dr. Ryan Hartman from the Department of Chemical and Biomolecular Engineering at the NYU Tandon School of Engineering. An associate professor, Dr. Hartman got his PhD from the Chemical Engineering Department at the University of Michigan. He did his postdoc at the Chemical Engineering Department of MIT, and also holds an undergraduate degree from the Michigan Institute of Technology. He has received quite a few accolades, including NSF Career Award and a member of the National Academy of Inventors. He joined NYU Tandon after being an inventor, a founder, and working in the industry at Schlumberger Limited, if I pronounce this. Schlumberger Limited. My Greek accent always fails to follow up. And he's appointed to the he's also appointed to the Faculty Senate Council of New York University, the Faculty Executive Committee of NYU Tandon. He has also served in various executive leadership roles for the American Institute for Chemical Engineering, President-elect and member of the Executive Board of Directors of the International Symposia of Chemical Reaction Engineering, and the member of the Advisory Board of Reaction Chemistry and Engineering, a Royal Society of Chemistry journal. Today, Dr. Hartman will talk about data-centric microreaction engineering for a sustainable future. Let's give a warm welcome to our speaker today.
Thank you very much for the kind introduction. You know, it's not all that common that we give seminars on our own campus, right? So it's just a special opportunity, and I appreciate the opportunity to talk to my colleagues, but just to keep the jokes in the back at a minimum, okay? For those of you who know me. So I'm an expert in designing chemical reactors, and most often cases we're interested in designing chemical reactors to understand mechanisms of chemical reactions. And then we use that to develop catalysts. And usually the applications revolve around materials or chemical manufacturing, and things are evolving to the point where we're generating lots of data. And most of what we do now is data-centric or data-focused. And for the last few centuries, chemical engineers have been part of the problem. We developed chemical processes that generate a lot of waste, but now the focus has shifted to trying to figure out how to make more sustainable processes. So in that context, that's really a lot of what we do for the last 300 years or so. And if you've ever gone to take an organic chemistry lab, a very versatile tool that you've used is Erlenmeyer flask. That's the way that synthetic chemists have been discovering compounds for various applications for a very long time. But in the last decade, we questioned whether or not performing the reactions in the laboratory scale in flow is more efficient and can yield more accurate information for predictive models that you can then use to scale up to actually produce chemicals. And so I know not everybody's a chemical engineer in the room, but if you're using a flask like this, if there are other rate processes that are limiting, like heat transfer or mass transfer, that can mask the actual kinetics of a chemical reaction. So then we use small scale flow systems to get information faster, and also I'll show you in a minute, it's more efficient in terms of energy. We reduce the volume of chemical waste, so we don't worry about disposing of so much in chemicals. And in some cases, it's even safer if you're dealing with hazardous compounds that could, for instance, be a danger to your health. So I'm gonna touch on 3 areas in this talk of applications that we've been working in for the last several years. The first is material synthesis and processing. And then the second is polymerizations in their life cycles. And then more recently, there have been some concerted efforts towards electrification, and we're working on a couple of centers at NYU focused on that to try to change the way that we actually drive chemical reactions. Okay, so those are not familiar with what microfluidics are. So small scale, fit in the palm of your hand devices. You flow fluid in, you can make different geometries, you can make them out of different materials, you can even make them out of silicon, like you make computer chips, or you can make them out of glass, You can make them out of fluoropolymers. There are many different ways to make a microfluidic device. And those are devices that have micro scale cross-sectional dimensions. But we're talking about flow systems. So we flow the chemicals in, we flow the chemicals out. We're interested in integrating these kinds of devices with in-situ analytical methods. And so these are just some applications of doing this. But Raman spectroscopy is a common technique used to measure different chemicals present. So if you take a laser microscope, integrate it with a device like this, you can see what's going on inside in real time. Another example would be, let's say you use an infrared camera or IR thermography. In this example, there was a reaction that generated heat. So you could see the temperature change in the system, you know, like the movies where you have heat vision. Well, in this case, we're using a camera. And then more recently, we're looking at using optical probes, plasma microreactors that I'll get to at the end of the talk. But those are just a few examples of kind of the thing that we do. So I mentioned that batch-wise manufacturing has really been the way that, that folks have focused on developing chemicals in industry as well as materials. This is just an example. If you went across the river to New Jersey to, for instance, Bristol-Myers Squibb, and you look at how they make different therapeutics, you would walk into a facility like this that houses these large-scale semi-batch reactors, and they take up a lot of room. But this space of using microreactors has progressed considerably in the last couple of decades. So the green dashed line highlights kind of where we work at the laboratory scale. But these small scale flow reactors provide information, like I said, for predictive modeling in the laboratory that we can then use for full scale production. So the batch reactor I just showed you, imagine replacing that by a bank of reactors that look like a computer server. It takes up a much smaller footprint, and there's also other advantages to doing that. So this sort of area is taking off, and I should mention that places like Southeast Asia right now are actually shifting all to continuous manufacturing of their materials and chemicals, primarily because they've had in the last decade a lot of accidents where people have lost their lives. But just think about this in that context, continuous flow reactors and the impacts that might have. In more recent years, we questioned whether or not you could take these kinds of systems and start to analyze the data in real time using machine learning algorithms. And so then I started working on machine learning, I think it was in 2013, even before I arrived here, because at some point, if you're in the lab running experiments with these small scale flow devices, you start to generate an incredible amount of information. You're spending much longer analyzing the data than you are actually running the experiments. So why not use computers to actually analyze it? So we've looked at artificial neural networks to perform a limited set of experiments. So let's say we only perform a handful of key experiments rather than a large number, and then we can sort of piece together the missing information. Of course, if we get too good at that, then the graduate students disappear. If we develop autonomous laboratories, but the happy medium is somewhere between that, and we can't seem to get enough funding from NSF to do what we really want to do, which are cyborgs, right? Like this $6 million for those who are familiar with that show from the 1960s. Okay, so back to the application. Imagine these large-scale reactors. This is a site, an aerial photograph from Eli Lilly in Ireland that was presented in 2016. All the blue areas highlighted in blue are these large-scale reactors that I showed you a picture of, and they're manufacturing drug compounds using this kind of an operation. Around that time, they designed their first flow facility, and it's highlighted in the green. So you have a 90% reduction in the footprint of manufacturing the same pharmaceutical compounds. You know, buildings are among the largest of CO2 emitters, and they require lots of energy because of the way the HVAC systems are designed. Not only that, the amount of liquid holdup, so the chemical waste that you generate is significantly reduced. So this is really an enormous impact. Just by designing reactors continuous flow, you significantly reduce the amount of environmental impacts in chemical manufacturing. Now, taking it a step further, if you talk to companies like, you know, Dow Chemical or even ExxonMobil or Sabic, these are monster polymer companies. You know, everywhere you go in the world, there's plastic, right? So these companies, what they do is they screen hundreds of catalysts to try to figure out the optimal conditions to manufacture polymers for plastics. If you can perform a few experiments like I showed you using small scale devices, At the same time, analyze the data with artificial intelligence, you reduce the time and the amount of chemical waste is generated by orders of magnitude. So instead of taking a year to screen hundreds of catalysts, you're talking about maybe weeks to months. Instead of generating tons of chemical waste, you generate maybe kilograms of chemical waste. So the environmental impacts are massive. So what I want to do in the time that I have today is sort of investigate three different fundamental questions that we have asked over the last few years. The first is, can we design microfluidic devices with in-situ methods to push beyond what we thought were possible for the discovery of material science? And I'm going to highlight three different problems we've been looking at recently. The second is really, what is the appropriate architecture for computers to work synchronously with high-throughput experimentation? And I'm going to talk about one example, which is catalytic olefin polymerization, because that's how plastics are made, which are virtually everywhere. And then the last, if you can see it, there's this text at the bottom, is, does the design of plasma microreactors hold promise for electrification in chemicals manufacturing, which is focused on more of the efforts that are going on in DCUs and the other centers that are working on at NYU. Okay, so I teamed up with my colleague, Bruce Garretts, who's also in the chemical engineering program a few years ago, because he had discovered in the 1990s, if you take a supersaturated liquid and you expose it to an optical field with a laser, under the right conditions, you can actually induce nucleation and then crystallization of compounds. So we thought this was interesting in that it might have application in chemicals manufacturing. It turns out crystallizations, especially in the agroscience industry, are among the worst when it comes to generating chemical waste. You're talking a massive amount of chemical waste is generated. If you can control the crystallization instead using a laser and minimize the amount of solvent that you use, it's a greener path forward. That's one problem I'll talk about. The other is the way that nature stores natural gas is in the form of what are called natural gas hydrates. So if you go to the Arctic and the permafrost, or if you ever have the opportunity to take a submarine down to the continental shelf in the ocean, nature's way of storing natural gas is in a crystalline form that looks similar to ice. So you get these methane molecules. That's what these spheres are here. They're trapped inside water molecules that form hydrogen bonds. And there's twice all other fossil fuels combined around the world stored in this form. So that's important, right? Of course, because if the natural gas is released, natural gas is a worse greenhouse gas than CO2. But in terms of energy technology, there could be applications there as well. The third application I'll talk about is related to conventional energy production. Asphaltenes are the highest molecular weight fraction of crude oil. And it turns out when you're producing oil and gas, they self-assemble into fractal structures or nanoscale fractal structures in porous media and then plug the oil and gas wells. So if we can design production more efficiently, it actually is a path forward to reduce the carbon footprint of current energy production technology. We looked at DNA origami folding. That's why this is here. A few years ago, we were interested in building a program on that with some other universities. I'm not going to talk about that today, but if you want to talk to me later, we have some papers on that. Okay, so if you take one of these microfluidic devices that I talked about, one of the advantages is that the length scales are a few hundred microns. You're talking about, something that's smaller than a millimeter in thickness. If you put that in contact with a Peltier or a thermoelectric module, and then you apply some voltage to it, you can heat and cool these systems within a matter of seconds. So you can control the temperature. If you look at using an IR camera from the top down, you can create what are called isothermal conditions. So the temperature is the same everywhere inside this. So it becomes a model technique to perform experiments in the laboratory. And it turns out to be very useful for gas hydrates. So if you flow in methane in this case, which is the largest component of natural gas, and then a thin layer of water along the wall, this is what's called an annular flow, then you can get crystals forming at the interface between the liquid water and the natural gas. And my former student did that. This is what the device looks like. So it fits in the palm of your hand. And if you cycle the temperature using the thermoelectrics around the phase boundary for this crystallization, you can control the crystallization in a matter of seconds. Is there a question?
No, the software.
Oh, sorry. Yeah, my computer has a mind of its own.
Yes.
Okay, so at this time, this was the first example of being able to control quantitatively the crystallization of gas hydrates. Many people for like 50 years have studied gas hydrates using large, high pressure batch cells. And so we eventually got the attention of the world experts in this area, Carolyn Coe and Denny Sloan at the Colorado School of Mines, and we're now collaborating with the DOE I'm going to talk about in just a minute. But that allowed us to control the temperature within a matter of seconds around the phase boundary. And then my former student, Weiqi Chen, who now works at JSTAR, which is a collaborator with Merck, he basically, what he did is he looked at how fast the crystalline films propagate. So it might be difficult to see, I think, from this image, but in the paper, it's more clear. You have this hydrate film that's forming at that liquid gas interface, and it grows axially. So how fast it grows are the crystallization kinetics. And with in-situ Raman spectroscopy, you can hit that crystalline film and confirm that there are peaks that correspond to the hydrate structure. Now, what's interesting is if you can form the crystals, then you can then melt them, or what's called dissociation, and release the natural gas. So there's like energy technology applications, right? If you imagine, if you want to store natural gas, you could form the crystals, transport it safer. It's not high pressurized gas, it's just a solid. And if it catches fire, it's just a slow burning crystal. So in that case, he looked at the dissociation, and then with in-situ Raman, you can see the methane peak rising. That's because methane gas is being released. And then the peaks that correspond to the crystal are depleted. Now, this all happens in a matter of seconds. Okay, so long story short, not to bore you with all the theory, what we were starting to look at was how fast is a hydrate releasing the methane compared to how fast the heat transfer resistances are in the system, which has applications. And so after that, my other student came along, Tieni, who's now at Cornell, Tieni Hua, and he started to think about, you know, gas hydrates in the context that they are in the Arctic permafrost as well as in the deep ocean. But it turns out in the real world, they're part of an ecosystem and they're stabilized by living organisms. So we published this paper where we started to look at the gas hydrate inventory, and we're talking about gigatons of methane. So like I said, twice all other fossil fuels combined. So you're talking about a massive amount of methane stored in the form of hydrates. And depending on how deep it's in the ocean, it actually is stored in a different way. So the deeper you go, the hydrate crystals are actually in the formation itself. In shallower depths, they're actually in the surface and you can see them. So if you take a submarine, you can see these ice formations. And in those shallower depths, there are these worms. They're called feather duster worms that live in close proximity. And they're ancient bacteria that feed on methane as well as form methane. Well, it turns out the worms, in this case, these feather duster worms, they actually eat one type of bacteria, so they're predatory. But then the other type of bacteria, like I said, forms the methane. So you get this symbiotic relationship. And that was interesting to us because we wanted to understand better what the energy balance of the system was. So methanotrophs actually consume the methane, and that's an exothermic process, whereas methanogens form it, and that's endothermic. So if you take away the heat generating microorganisms, because the worms are eating them, that can shift the amount of heat that's generated. Think about that in the context of what I showed you on the previous slide. The hydrates can actually melt if you add heat to them, right? So depending on the system, if there's a large population of these worms, or if the worms are non-existent, you can reach critical temperatures of the gas hydrate films where they start to release the methane into the ocean. And that's sort of what we study. We don't really know what would happen to the methane after it's released because it might just be carried away by currents and then form crystals again. Or it actually could rise, I suppose, to the surface of the ocean. We're not sure about that, but that's a separate part of the problem. So around that time, I got together with Carolyn Coe at the Colorado School of Mines, and we started asking questions about, you know, if these hydrates are forming in nanopores, let's say they're deeper in the ocean, away from the worms, and they're actually crystallizing in nanopores. Does the local pore geometry influence the crystallization kinetics as well as the stability of these hydrates? And that's what my postdoc, Mrityunjay Sharma, is working on with, sponsored by the Department of Energy. And so then we're trying to understand different shapes of nanopores as well as the chemistry of the nanopores and so on. And so that's really the next step on the hydrates. Like I alluded to earlier, Bruce Garretts and I are working on what's called non-photochemical laser-induced nucleation. And in that case, we've integrated a laser with a microfluidic device, and then you can easily flow the fluid through the path of the laser and then use a high-speed camera to actually count the number of crystals that form, look at their shape, look at their size, and in some cases, you can get information about polymorphs of the crystals. So this is really useful, especially for pharmaceutical companies where maybe you're interested in a particular size of the particle or a polymorph, and that's really your active pharmaceutical ingredient. At this point, Bruce and I are questioning what is the mechanism, because we still don't understand, of the light inducing nucleation in the system. And one of my students, Kalichi, is working on this to try to understand. There's actually three mechanisms, one's missing from here, but if you have nanoparticles in the system, the light can cause the formation of a vapor phase around the nanoparticle, and that could lead to nucleation. Or it could be that the actual laser itself causes some alignment of the molecules, and that's called the optical Kerr effect. And then there's also what's called the dielectric model. So one more slide. Before I get to that, like I said, you can measure the particle sizes if you have a high-speed camera in situ. So there's also what's called the dielectric polarization model. And my former student, Tianyi Hua, actually showed for simple systems like potassium chloride and glycine that you can predict the mechanism. This is just the lability that describes the nucleation kinetics for that particular model. So I don't want to get lost in the terms here, but you could measure the number of crystals using the camera, correlate that to the properties of the laser, and then work back what the mechanism is to see whether or not you can predict what the kinetics are for the nucleation. So why are we using microfluidics? Because it allows us to quantitatively measure the crystallization process and correlate that back to the laser characteristics, which is really the first time that anybody's done that. Normally you use a batch system. If you shine a laser through it, all the fluid inside that batch system is not going through the path of the laser, right? So you use probability theory to figure out what's happening. More recently, and what we're looking at now are other types of molecules that are more interesting and relevant to pharmaceuticals as well as fine chemicals. But just another application. Those who work in the field of material science often question whether there's more than one activation barrier in the mechanism of nucleation. And so we're actually starting to look at whether we could use microfluidics with a laser to work out if there's two-step nucleation. In this one paper we published for glycine, we showed that there is two-step nucleation, or we think there is. Okay, so the last part of this, the first talk, is just this application of asphaltenes. It must have been in 2011 or 2010, I started talking with oil companies about some interesting problems to work on. And asphaltenes are actually more like fine chemicals and pharmaceuticals. They're highly aromatic molecules in the highest molecular weight fraction of crude oil. But we started to look at, could we rethink how we study asphaltenes using microfluidics? Because up until that point, even my lab, when I used to have an industry, the techniques to study oilfield chemistry were outdated and they're costly, time consuming, and wasteful. So if we can use microfluidics to speed up everything and design more efficient field operations, you reduce the carbon footprint right away. You reduce the amount of natural gas that's leaked off and all these other problems that we've heard about in the news, right? So we eventually progressed to the point where we designed automated microfluidic systems. And the reason we wanted to do that is because there are so many different chemical interactions of these molecules. And imagine if you have an oil well, you drill this well, you have a porous media where the oil and gas is flowing through. These asphaltenes undergo these chemical interactions inside the porous media. So it's not like they're just in an open medium. They also have surfaces to deposit on. And if you ever go, into your backyard, if you have one and start digging, and you analyze the soil, it's incredibly complex in terms of the mineralogy and the inorganic chemistry. So this is really a massive combinatorial problem. And my first student, Chun-Tien Hu, looked at this, who's now with Continuous Pharmaceuticals, is this MIT spinoff company. Basically, imagine, a microfluidic device with sand particles in it. So these are ground quartz particles. Then we could precipitate out these asphaltenes, and that's why from left to right, this packed bed gets darker. And then you can measure the pressure drop across it to show that you're actually plugging the packed bed. Then I had a postdoc that came along, Bruno Pinho, who's now in the faculty at University of Cambridge in chemical engineering. And he took Raman spectroscopy and created a 2D grid of this packed bed. So every one of these green points, you're creating a spectra. So we got tens of thousands of spectra that are being collected. And you can analyze that in real time if you use MATLAB. And you can deconvolute that spectra to figure out how big these asphaltene molecules are, but also where they're located inside this packed bed. So this was like useful because we could see now where are these asphaltenes depositing in this microfluidic porous media. So all the red is where they're depositing. And we could change the surface chemistry of the packed bed. And then we could also measure on the 8th floor of Rogers Hall, which is like 12 stories above the subway while the whole building is shaking, the size down to a few nanometers, which was exciting for us. So that was a new technique then that we could consider for this problem of asphaltenes. But we took it a step further, and this is kind of where we left it. If you want to get rid of them, you inject different chemicals. So if you don't do a good job designing what's called a squeeze treatment in field operations, if you inject the chemicals and they don't go where you want them to go, they're not going to dissolve away the asphaltenes. So we could start to inject chemicals and look at where they're also at inside this packed bed. So just to summarize, there's lots of interesting applications in material science with microfluidics. We started to look at gas hydrates, and it's really led to some interesting opportunities to rethink and measure the kinetics of crystallizations in this area. I'm working with my colleague Bruce Garretts on non-photochemical laser-induced nucleation because we don't understand the mechanism. And we think that using microfluidics, we can sort out the mechanism. And then finally, in the space of asphaltenes, this is more of a soft matter problem. If you're interested in self-assembly inside of complex structures, microfluidics with in-situ spectroscopic techniques are useful for that. Okay, shifting gears. I talked about plastics. Plastics are everywhere. It's a problem not only on the front end because manufacturing polymers requires a lot of chemicals and energy, generates a lot of CO2, but some of you may work in adjacent areas on the downstream side. Plastics, what do you do with them after you're done with them, right? I'll come to that at the end, but this mechanism, which I certainly don't expect you to understand, this describes most plastics, the way polyolefins are polymerized, but we are interested in this because as a chemical engineer, I can model this and then try to look at how different catalysts influence the mechanism and how long it takes to polymerize. And so it turns out these polymers are made using metallocene catalysts. So at the bottom here, this is just an example of what a catalyst looks like. And so it's a zirconium-based catalyst. But back to the the societal impacts of this, I think my student came up with a number, roughly 6% of all energy produced in the United States is used in the production of polymers. That's a massive amount of energy, and then 37% of all greenhouse gases are directly or indirectly generated by polymers manufacturing because these come from ethylene, from natural gas wells. And so this is a massive sustainability problem. These chemical reactions generate heat. So as the polymerization happens, it heats up the system. So imagine using a microfluidic device like I talked about, but looking at it using an IR camera, so infrared camera. So that's what Benjamin figured out. And he took it a step further. If you then take images in this way using an IR camera of what's going on in the microchannel and train an artificial neural network, then you can actually control the temperature of the device in an experiment. I'll get back to that in just a second. Some of you may already be familiar with what artificial neural networks are, but you can have inputs and then hidden layers or neurons that are biases. They have biases in them and weighted functions depending on what you're interested in fitting, and then outputs. And from a process dynamics and controls point of view, this allows you to do what's called feedback control of a process. It's actually been around for quite some time in the space of chemical engineering. We've taught this in our process dynamics and controls course for a few decades. But if you take an image of a reactor where this polymerization is happening and it has some color pattern in 2D, and you analyze it at the maximum voltage applied to this thermoelectric module, then you take another image at the lowest amount of voltage, so no voltage applied, and then you take random set points in between using like a design of experiments algorithm. Then you can train an artificial neural network, store it in a database, and then turn it around and use that to actually control the temperature of a microfluidic device. So why do I care about this? Well, look at the temperature response as a function of time in this graph in the upper left. So the artificial neural network is controlling the temperature of the microfluidic device by applying voltage to this chip, and it either decays right to a set point or it ramps right to the set point within a matter of seconds. The way that we usually control the temperature of a process is by what's called a PID controller. And you get overshoot, decay, and then finally at some point it reaches a steady state. This is how you control the temperature of this room using a thermostat, right? That's why it's hot sometimes, cold sometimes, right? If you want to run thousands of experiments in a day, or maybe a few days, and screen hundreds of catalysts, time matters, right? So if you can control the temperature of this device within a few seconds, then you can shift from one set to the next, to the next, to the next of operating conditions. So that's why we did this. Taking it a step further, once we figured that out, we injected the reagents and carried out the polymerization. And this is what it looks like in the lower left, where the color changes. And then we can take the images based on how much heat is generated, train the artificial neural network based on what the kinetics are, and then start to look at things like the reaction space topology. Okay, a side note before I get to that. If you've got a system like this and you're generating data and analyzing in real time using machine learning, you're talking about generating terabytes of information, which isn't realistic to use a hard drive like in your lab, right? Unless you've got a bank of servers, maybe you do, I don't know, I don't. So what we did is we used Amazon Web Services, and that worked fine to do this, cloud-based computing. And then we had our system design software that was running the pump. So it was a fully automated system where we could analyze the data in real time. Our pumps are operated by the computer using a system design software. At that point, then we use an algorithm. Yeah.
So what is the data at the output that is so heavy?
Yeah. The images.
So if the picture, then
The picture.
that's heavy because a lot of pixels.
Yeah. So depending on the resolution of the camera, number of pixels, all these other things, you can really start to get into where you're generating an incredible amount of information.
And you need that all.
Yeah.
Oh.
Yeah. Now, okay, we analyze what's leaving offline. That's part of this. Some other people have analyzed what's leaving their reactor inline and analyzed it. Those data sets tend to be less intensive than an image like this. So you can probably get away with gigabytes of information rather than terabytes, depending on your approach. Okay, great question. So now if you tell your computer, I've got a catalyst and it's at some concentration, but there's a monomer that's involved in this polymerization and there's a co-catalyst, then you can do 30 sequential experiments using design of experiment algorithm like Latin hypercube, just to randomly choose combinations of those three to train an artificial neural network. Based on the output response of the images, then you can go back and predict the catalytic productivity for different sets of conditions. And that's essentially what Benjamin did, who then has since left to AbbVie, where he implemented this whole idea in Chicago. Now he's at a startup company. But the red shows you the predictions from the trained neural network. The blue is actually the experimental measured data. Why is this important? Because then you can start to map out the reaction space with only a few experiments. Remember back at the beginning, we don't want to run experiments all day long every day. Experiments are expensive, they generate chemical waste, and they take time. So if we can run just a few experiments and then analyze everything in real time with advanced data analytics algorithms, then I think we're reducing the carbon footprint. I just wanted to mention, this is supervised machine learning. And the reason for that is because we're telling it up front what the mechanism of the chemistry is. We already know, like I showed you at the beginning, the different reaction steps, and each of these have time-dependent constants. You know, people are starting to look at deep learning algorithms where they sort of go at it, you know, with a black box mind, but I don't know, I always felt it should be based on some physical principles, and that's built into this in a supervised machine learning algorithm. So like I said, you run a few experiments, then we go back and we predict what the reaction space looks like. So now we have graphs of the catalytic productivity as a function of the monomer, as a function of the catalyst, and we can look at how the space changes for different sets of conditions. And so instead of running thousands of experiments, we only run maybe 10, 30 experiments like I showed you in a matter of 2 hours. Now, one day, my student Benjamin showed up and he said, I want to do this inside of a box. And why do you want to do this in a box? You're not going to take this out of the lab. I'll get in big trouble, right? So imagine taking the chemical lab and putting it inside of a Pelican case with a robotic arm and the microreactor I showed you, and then working in the data analytics. So you design this system, I guess it was sort of an engineering exercise, but what he was interested in was studying the co-catalyst. So there's a catalyst involved, metallocene catalyst, but then there are also co-catalysts. And then he took this system and studied 9 different co-catalysts of varying chemistries. So if I'm a developer, say like I'm at Dow Chemical or Exxon or Sabic, and I want to know which is the best co-catalyst to optimize a set of conditions that maximize the productivity of my polymer, or perhaps molecular weight or other properties, then this kind of system you could unleash and start to map out what the space looks like. So these are just 2D representations similar to what I showed you previously, but look at the area in yellow. This is for antimony chloride, which is one of the co-catalysts. We see that with the lowest amount of monomer needed, we have the highest catalytic activity of this particular molecule. And if we went back and looked at the literature afterwards to see whether or not the machine did in fact figure out, and it did, it figured out which was the best co-catalyst to use with this system. So this is kind of a step towards autonomous laboratories. And to wrap up, since then, folks in the medical school have approached me about studying microplastics. The problem there being that the physicians are really concerned that microplastics are having some negative effects on our health, but it's very difficult to identify them, characterize them, and screen for them. So what we're doing is we're looking at a microfluidics-based approach with machine learning in order to study microplastics from different human tissue samples, not only from your blood, but also your lungs and even other vital organs. So that's kind of where it's going. So I talked about using IR thermography to control the thermoelectrics, so the temperature of this device, so we could do high throughput screening. But when you don't have much data, or where you want to run a few experiments, this is an effective way to analyze in real time to get the kinetics, and then rapidly screen for different parameters of your system if you've got more than one catalyst that participates in the reaction. That's useful. But all in all, I would say that applications of artificial intelligence in the space have a great future. There's just a lot of different problems to look at. This is just an example of one polymerization. Okay, shifting gears and then try to wrap it up. I want to talk about this energy transition part of the of chemicals manufacturing. This is just a news article that was published in 2021. So Dow invested in building the first net zero ethylene production plant in Alberta, Canada. And so net zero means the overall carbon output will be zero. And the way they're going to do this is by CO2 and sequestration. So they're going to capture any CO2 that they generate and then inject it back into the ground. So that's a key part of their technology. They're also designing into it the use of alternative fuels for cleaner thermochemical transformation. So they're going to use hydrogen to heat some of their process units. So that's an interesting approach. But at the same time, around the same time, I've been working with my colleague, Miguel Modestino, Andre Taylor, Ida Idle on our center, DC Muse, as well as another number of other proposals focused on electrification. So that's the other approach to this is imagine instead, you know, a future where you use electricity, instead of having to inject CO2 in the ground, can we use solar energy and eventually drive chemical manufacturing with for instance, just water or natural gas or even CO2. So I have this project with NYU Chemistry as well as the University of Michigan, where we're looking at methane activation catalysis in order to form fine chemicals and pharmaceuticals. And the way that we're interested in doing that is using plasma physics. So I'll get to that in just a minute, using plasma and what is that. But if you just break down and look at how natural gas is currently consumed, in 2019 numbers, most natural gas is consumed in pipelines and distribution, some residential and then other industrial. But if you look at feedstock sources for chemicals, overwhelmingly, we're using basically liquefied petroleum gases and others to manufacture chemicals. So to date, we haven't used methane all that much to make chemicals, but yet we have an abundance of it. You've probably read articles about flaring methane off, all around the world in landfills as well as in oil platforms. So why not use that with electricity to manufacture chemicals? We think we can use plasma, but there's a lot of challenges. So if you take natural gas like methane and you send it through a high voltage electric field, you break the molecule down into ions and radicals, and that's a plasma. Characteristic of that is a glow discharge. You'll see a glow. But there's all these challenges associated with plasmas. And so the intrinsic selectivity is one. Like I just said, you break it down into different species. So a distribution of species is not necessarily what we want. We want one species so that we can react it and then form a large volume or a quantity of what we're interested in. But plasma reactors, I think, require new innovative approaches because it's difficult to get high surface area with the plasma with reacting species. And I'll come back to that in just a minute. But energy efficiency is potentially a problem. You have to put high voltage electric fields. And so then depending on how you look at it, maybe you have an infinite supply of solar energy, and that is potentially in the future. But at the current moment, we don't. So energy efficiency is a problem. And existing manufacturing processes are fossil fuel driven. So the entire infrastructure on manufacturing chemicals depends on heating chemical reactors or cooling them using fossil fuels. But where we come into the picture is we don't have many techniques to study the catalysis or the chemical reactions at the laboratory scale. And that's really what we're working on, to try to understand how we can measure the kinetics and then screen different catalysts. Just as a last point, plasma catalysis is a relatively new field. Okay, so I talked about microfluidics at the beginning, right? We can design devices out of different materials of construction. It turns out if you deposit an electrode, you can create what are called dielectric barrier discharge plasma reactors. So imagine a glass microfluidic device. You have an electrode on top and bottom. In between the two electrodes, there's a dielectric barrier. The insulating material is glass, but electrons are still transported. So you can form in that space a plasma and then start to study the kinetics of different reactions. And so this is just one example. If you use an optical probe, this is called optical emission spectroscopy or OES. The glow discharge gives off light. You can detect that light and then look at where it shows up in different shifts of wavelengths. So then you can break down what different species form. And depending on the peaks that show up, then we can say we destroyed the methane and it formed a plasma or not, one way or the other. We've got a couple of efforts that are focused on studying plasmas in this way. The first is DC Muse, I mentioned, our Center for Decarbonization of the Chemicals Industry. And there we're starting to think about a heterogeneous catalytic approach. So if you take a particle that's a catalyst and you put it in this kind of an electric field with a plasma, can you carry out plasma catalysis in a packed bed? And this is a very common way in industry, minus the plasma, just use a packed bed. That's a common way for chemical companies to manufacture chemicals. The other approach that we're looking at is similar to the polymerization chemistry that I showed you. So imagine that the catalyst is actually a molecule that's dissolved in the liquid medium. In that case, then the catalyst actually flows in and flows out of the system. And we've got a project with the National Science Foundation through their EcoCBET program. And then I started looking at this with my NSF Career Award. So there's two fundamentally different approaches of catalysis to study plasma. So first is using solid catalyst. The second is using a liquid catalyst. And so I had a former student who designed a device, a microfluidic device to study plasma catalysis. And this just shows you from the top down. Imagine you have posts that are in 2 dimensions, and then you have these channels on the side. So then essentially what we have is a 2-dimensional packed bed, and we can flow the gas in from the sides, flow the liquid through that packed bed, and then generate a plasma to react with whatever liquid's flowing into this device. And long story short, not to bore you with the theory, but if you want to use a device like this to measure the kinetics, in chemical engineering, we often evaluate what's called the dispersion. So as the molecules flow through, there's some time scale for that. There's also a time scale for diffusion, molecular diffusion, and how fast that molecular diffusion is relative to how fast the molecules are going through can affect your models. And so my student in this case worked out a model that describes, this is basically a dispersion model that's well understood, and then we fitted it to figure out whether or not dispersion would be important. So we concluded at the end of this that you would have to account for dispersion, even if you use a small scale device like this. Why is that important? It's important because, like I said, plasma inherently has a distribution of species. So if there's a concentration gradient because of molecular diffusion inside this microchannel, and there's a distribution of species from radicals to ions and other components, you're talking about different reaction rates at different points axially inside of this device. So if you want to work out a kinetic model that would tell you the intrinsic kinetics, you have to account for those gradients. This is why we did this in the first place. But back to the device itself, like I said, you can put an electrode top and bottom, and then when you turn on the high voltage, you see characteristic shifts that correlate to the formation of a plasma. So you can measure and confirm that there's actually formation of a plasma just by looking at the electronics of the system. The other way to do it is, like I said before, is to look at a glow discharge. So this is a picture of the purple glow that you can see. So that's the second piece of information that would tell us we formed a plasma. And then the third is actually measuring using optical emission spectroscopy the shifts of the radicals that form. Okay, so before I wrap it up, because I think I'm probably running out of time. Okay, the other advantage of using a device like this is that you can create a high intensity electric field in a small scale dimension. So if you create very high intensity electric fields, which is if you plot the electric field as a function of power density, the triangle here corresponds to our experiments, and then the triangle here, and then also the circle. You can see that we're operating at high electric field strengths and power densities in the system. So that really allows us then to look at sort of selective conditions. If you scale up to a large scale reactor, it's difficult to get to these kind of power densities. But back to the story. So if you use optical emission spectroscopy, you can say, I formed these other molecular states of carbon hydrogen. So I destroyed this methane molecule that went through this device, and we've measured that we've destroyed it. And so if I'm somewhere and I have electricity and I have natural gas, I know I can form a plasma in a reactor like this. But what was really exciting is what we did with it next. So this is a carboxylamine, which is a vitamin B12-like complex. If you ever wonder what vitamin B12 looks like, cobalt is a transition metal that's coordinated to the complex, and there's a cobalt-chlorine bond in this complex. It turns out in your body, there are biocatalytic reactions that take place using this as a catalyst. So we thought this might be a good model molecule to look at for catalysis. So what we did is then we sent this molecule in liquid droplets through this reactor that we formed the methane plasma. And then we think we kicked off the chlorine, and what we had at the outlet was a methylated cobalt complex. So we did direct methylations with the natural gas and electric field with this vitamin B12-like complex. If a pharmaceutical company is interested in doing something like this, normally you would use an organomethane source, and there would be 2 to 3 other reaction steps in between. You would also have to carry out separations with solvent. And so you're talking about much more chemical waste and time in order to do the same thing. Now, what we really want to do with this next, we haven't done yet, but we want to take a complex like this and then react it with other molecules to form drugs. So the long-term vision is, can we use an electric field like this with plasma to form drug compounds with natural gas, which hasn't been done before. And so if you're asking the question, is that really a cobalt methylated group? We carried out some experiments to show that it is. So if you just look at the shift around 492, that shows up because we formed a cobalt methyl bond stretch in the Raman spectra. And then there's also a few other spectra that validated the formation of that complex. Okay, so I know I'm running out of time. We're interested in reactions of the plasma with liquid interfaces. So imagine the vitamin B12 in the liquid, but then that gas phase is where the plasma is. Creating a high interfacial contact area and one that's stable is actually a challenge when it comes to reactor design. And that's part of the problem we're looking at right now. So to wrap up, dielectric barrier discharge microplasmatrons is what we call them, can be made and then studied with high electric or high voltage intensity electric fields. And the advantage of that is that we think we can use them to measure the kinetics of important chemical reactions, where one might want to use natural gas in catalysis. We're progressing to the point where we think that we can eventually couple methyl radicals to interesting complexes that are pharmaceutical-like, and that's part of this program that I have with EcoCBET and NSF. But we're also starting to look at heterogeneous catalysts, which I didn't talk much about, with DC Muse, which is the center. So to wrap things up, there's just a number of people that have been involved in this research I'd like to thank. Jasmine was one of my former students who worked on the plasma reactor, Karishma on the asphaltenes, Benjamin on the polymerization and the AI, Luigi on the asphaltenes, as well as Priyangi, and then Bruno on the analysis of the, as well as the gas hydrates, but the analysis of the packed beds, and then Tieni on the gas hydrates. And none of this would have been possible, of course, without funding. So we've been fortunate to work with NSF as well as the US Department of Energy and Anadarko Petroleum and NACO Champion. And the gas hydrate work that we're working on now, actually, it started years ago with Chevron in a project I had because they're interested in gas hydrates forming in oil wells. But more recently, when I came to NYU through our MERSEC on the main campus. So with that, I'd be happy to answer any questions.
So we have roughly 10 minutes for questions. We're going to end around 1:05, not around, at 1:05. So any questions? I will be here just to repeat, or if you want to repeat, up to you. I have one question until oh, go ahead, Jackie.
I was curious to hear more about, you mentioned in one slide about the work you're doing in the medical center on looking at microplastics in the human body and then using machine learning for that.
Yeah.
Could you maybe just go over a little bit like what is the machine learning used for there? What are they doing this analysis?
Yeah.
So just to repeat the question, so we have it on record. What are the inputs and outputs of the machine learning methods used for
the medical microplastic research?
The medical microplastic research.
Yeah. So the major challenge that we're having is that when you process a tissue sample that has, we think, microplastics in it, there are only a few particles per mass of tissue that are processed. And so then the physicians are dissolving cadaver samples for us. And then we're taking them and filtering them at the moment and then looking at them under a microscope. So that's like searching for a needle in the haystack, if you want to call it that, which isn't very efficient. So I've got students designing instead a microfluidic approach where you take that sample that the physicians have processed already, flow it through a device, and you can use hydrodynamics to focus the particles. So without getting into the specifics, but there's a paper here at the bottom. If you're interested, I can send you. you can send in a liquid sample with these particles. With the design microchannel, you can focus them by the time they leave into a sort of a chain or a flow path. Then if we take that and we analyze it using some in-situ spectroscopy, that could be a microscope, or what we're doing is using Raman spectroscopy, then you can either count them or you can look at their characteristics one at a time. So the inputs and outputs of the machine learning would be well, basically what we start with here, whatever the characteristics are of the process sample, and then what we measure using the Raman microscope. So that creates our feedback loop. Now, I can't say too much more about the actual algorithms that we're using at this point, but to give you the whole idea, if we do it this way, we're not searching for a needle in a haystack anymore. We're forcing the particles to go where we can measure them. So we don't have to search for them. Does that make sense? And this could be really powerful, we think, in terms of, let's say, the physicians want to screen many different types of tissues and different types of conditions that they think are resulting from microplastics.
So the idea is that the model you're trying to learn, if you're allowed to say it, but is to how to get that single stream in the best way.
It would be, once we get the single stream, how many particles, what are their size, what's their chemical composition, can we relate the characteristics of the particles, including how dense they are in the body, to basically the diseases that the physicians think are resulting in death at the front end.
So it's what you're doing with that in-situ spectroscopy step. That's the machine learning part.
Yeah, that's right. That will be basically part of our algorithm.
Thank you. There was an amazing talk. Thank you so much.
Yeah, you're welcome. I think this actually, not just microplastics, there are other types of problems like this that this could apply to, not just particles being found in human tissue, but we have samples already just using the method I talked about, where we're searching for a needle in a haystack, where we have found plastics in human tissue, which is alarming. It's alarming.
Thank you. Go ahead.
Great, all right. I always wonder, is there a possibility to lower the energy consumption of these plasmas? Like, you have a lot of energy in a very small volume. I assume most of it is going to heat. Are there strategies to minimize that amount of energy that is lost? Or?
Yeah.
So question is, are there strategies to minimize energy consumption in the creation of plasma?
We go to the schematic okay, here. The question is really, if we're applying an electric field across this device, and this is a dielectric barrier, inside here is where the plasma is forming. What's the efficiency of this? Because you're talking about, 10,000, basically 10,000 volts, So this particular device, we had to calculate everything to satisfy our safety folks, right? It came out to be, I think, 75 watts, so like a light bulb of power, but it's not really scalable. And so the problem is that we don't have right now a way to measure inside what the actual potential is. And we've discussed this back and forth. So our group at Michigan is simulating it and trying to predict, and we're definitely below 10% efficiency. So I think the answer is yes. It's just that's like several generations of iterating reactor designs, and hopefully at some point we can measure inside the device to match whatever they're predicting. Now, there's different types of ways to form plasma. This is what's called a dielectric barrier discharge, where you've got this insulating material between the electrodes. So inherently, you're losing, right? Your efficiency is going down because of that. You could also take an electrode, put it inside of the flow field, and generate the plasma that way. And that's what actually our collaborators are doing at the University of Houston. So it's a sort of a different approach. But that could lead to some interesting questions about the catalysis, because that's the other part of the problem. When you're designing a packed bed, right, to carry out plasma catalysis, well, even if you have the electrode in the flow path you have all these solid particles. And so then those also create resistances, right? And so the dielectric barrier might not be all that far off from actually what we will be dealing with on a larger scale when we want to do manufacturing. But we're not there yet. And hopefully we can measure what's inside and then validate what they're simulating. Our collaborators are simulating.
Thank you, Dr. Hartman. Let's give one more round of applause to our speaker today,